So I'm going to be talking about a methodological problem which came up as part of a metronome synchronization task which I was using as part of my PhD research on prosody in Munster Irish, one of three dialects of modern Irish. As part of this investigation, I was looking at a possible relationship between perceptual center or P-center location and initial consonant mutation, which is a morphophonological process which exists in modern Irish. So a methodological problem came up as part of this, and I'll be discussing that rather than the study itself. So I suppose I should start with what the talk is not as part of this purpose and structure. The talk is not about P-centers themselves, although the concept of P-centers are relevant. And likewise, it's not a talk about the study itself and its findings, although this is interesting and I'm happy to go into this either in the Q&A or personally after the conference, but rather these things are just relevant context for a particular methodological problem which came up as part of this process. And this is practical information which would have been useful to me when I was planning my study, despite having surveyed the literature quite extensively. So hopefully it may come in useful for other researchers as a small practical note on experimental methods. I'll begin by going through some relevant background information on initial consonant mutation and on P-centers. I'll then go through the first iteration of the task in question, which was a pilot study, or took place as part of a pilot study rather. I'll then go on to the second iteration of the task, which took place as the beginning of what was supposed to be main data collection, but was derailed slightly by this particular problem which came up. I'll outline the problem itself, and then I'll propose a solution and make a few concluding remarks. To begin with a bit of relevant background and context, then, I'll start with initial mutation, which is the morphophonological process which I was investigating. Initial mutation is an umbrella term for a family of phenomena which occurs globally, although it's typologically quite rare. It roughly refers to grammatical alteration of the segmental character of a word's left edge for various grammatical purposes. In Irish, it's used to signal grammatical gender and number and tense, amongst a few other things. There are two such processes in Irish. Other Celtic languages may have up to three. The first in Irish is called chevu, or its traditional name in English scholarship is lenition, which was historically a spirantizing process, which produced roughly the alternations which I've laid out on the slide there, although it's no longer straightforwardly spirantizing, and even what I've laid out there is a bit of a simplification. The second, and the one that I'll be focusing on, is called uru, or eclipsis, which was historically a nasalizing process, but is no longer only nasalizing. It also voices stops and F, in addition to nasalizing voiced stops. I ended up focusing on eclipsis, one, because its alternations are a little bit simpler and straightforward and subject to less variation in realization from person to person and from dialect to dialect. And it's also a little bit more straightforward for controlling for segmental structure in words which are available in the lexicon, so it was easier to control for a variety of factors. And perhaps most importantly, these mutations, especially uru, with very few idiomatic exceptions, can only occur in the context of a short phrase, because you need a grammatical particle to trigger the mutation, and then you need the item on which the mutation is triggered. And this is the reason why, unlike most other studies involving metronome synchronization and P-centers, I couldn't just use a single word or a single syllable. I had to use at least two. So this is probably why this hasn't come up in the literature before, to my knowledge, and why it's useful to have learned this fairly early in the experimental process. The second relevant concept is that of P-centers, or perceptual centers. There's an awful lot that can be said about P-centers, and an awful lot that has been said about P-centers, and an awful lot that I won't be able to say about P-centers in this time or in the scope of this topic. So I'm going to grossly simplify, with apologies to anyone who may have better expertise on the topic than I do. It's a concept which was originated by Morton and colleagues in 1976 to account for issues in perceptual rhythmicity, the perception of sequences of syllables as rhythmic, in a psychological experiment that they were carrying out. It's been referred to as the perceptual downbeat of a syllable, or the moment of perceptual occurrence of a syllable. It's sort of when a language user's brain, if I can say that, clocks that a syllable is happening. You'd think this would be very straightforward and would have to do with the onset of acoustic energy, and there's a link to that, but it's not quite as straightforward as you think, as I've very roughly schematized in that illustration at the bottom of the slide. in which the alignment of what you would think of as straightforward acoustic energy, the beginning of syllables, in this case the digits 1, 2, 3, 4, if you align those regularly, the percept is not a regularly spaced sequence. It sounds very arrhythmic, so the thing which has to be spaced regularly is what's called the B-center. There's a number of ways to examine this, depending on whether you're interested in its acoustic correlates. It is still fundamentally meant to be a psychoacoustic phenomenon, so there is an acoustic basis, even if it isn't straightforward. But if you're looking at higher level linguistic influences or potential influences in the location of the P-center, metronome synchronization is a very common paradigm, experimental paradigm, because it's easy to implement, it's easy to train your participants onto, and it's fairly straightforward in analysis. It does what it says on the tin. You play a metronome beat to your participants and present them with stimuli, and you direct your participants to then repeat the stimuli that they see in time with the metronome beat. You then analyze where the metronome beat has fallen relative to segmental material, depending on various criteria that you may determine based on the language that you're studying or any other number of criteria. And then the common stipulation is that where the metronome beat has fallen relative to roughly probably the left edge of your syllable is what you'll take to be the P-center, accounting for a standard error of some sort. The thing which has interested me the most recently in the literature is that there's been a proposed link with morphophonology, experimentally supported by work by Catherine Franich in 2018, who was working on a process of initial prenasalization in a Grassfields Bantu language called Mdumba, in which she found that homophone pairs with initial prenasalization differed in their P-center locations, apparently based only on whether the member of the homophone pair was morphologically complex and the prenasalization had been derived, or whether it was morphologically simple. This is interesting to me as this is fairly similar, although not identical, to what would have been the pre-morphologization stage of initial mutation in the Celtic languages before they became these more opaque grammatical processes. So I decided to have a closer look as part of my PhD. The first version of this task took part in a pilot study, which was very small. I had 3 native speakers of Waterford Irish, which is one of 3 sub-varieties of Munster Irish, the southernmost variety of Irish, of 3 macro dialects on the island. The participants were played a 60 beats per minute metronome beat over-ear headphones in a sound attenuated booth in our lab in Dublin, and they were presented with stimuli, one of the 27 stimuli in the table on the slide, one at a time on a computer monitor, and they were directed to repeat these stimuli, whichever one it was, in time with the metronome beat that they could hear until I changed the slide, which I would do after 10 repetitions. I cast a very wide net of stimuli, largely in the hope of, well, first of all, in the character of a pilot study, but second of all, in the hope of giving as much of an opportunity for interesting patterns to emerge in all the different contexts tractable within the scope of this study in which this particular mutation can occur. So all of the more complicated stimuli were based on one of 5 monosyllabic lexical items, which were structurally controlled, which are listed in the second column of the table on the slide. These all had long vowels and could either begin with a simple onset. I stuck with a bilabial place of articulation for phonotactic reasons. And in addition to the simple onsets, complex onsets could occur in which you'd have one of those consonants followed by the velarized lateral rather. These items could then occur in a possessive phrase, either with the eclipsing mutation, which signals plural possession, that is to say their X, or without any mutation, a 0 mutation, which signals feminine singular possession. So you could have her X as opposed to their X. This could then be further embedded into what's called the dative eclipsis, context, which is just a definite prepositional phrase. So you could have on the X, or you could further make this into a possessive phrase and have either on their X with the eclipsing mutation or no mutation and have on her X. Despite having this wide net of stimuli, it became clear fairly early in the analysis process that this particular subset that I've circled was the one that was going to be the most interesting, or perhaps not the most interesting, that may be a mischaracterization, but it was the most straightforward in terms of coherence of participants. There was very little variation in how they treated this particular subset because they were not particularly complicated and they were fairly easy to analyze. They represent the smallest or simplest context in which this particular mutation can occur with a possessive trigger. And they're also completely segmentally controlled, unlike the complex onsets. So they form pairings of either lexical matches or complete homophone pairs. And this is very useful for looking at the one item that I was, or the one factor that I was interested in, which is to say presence or absence of mutation. And I could then look at the p-centers from there. Results were analyzed in Pratt. I've given an example spectrogram there in which metronome beat location I've indicated with red arrows, and the metronome beat location was then calculated relative to segmental material, such as the beginning of onset closure, the release of onset closure, onset of the nuclear vowel, etc. Consistent with expectations, metronome beats were largely placed within the closure for the onset consonant, which is roughly where you expect the p-center to fall. So I was able to calculate p-center location for comparison across participants and across contexts and across onsets of different duration by calculating metronome beat location as a percentage of onset closure duration. Now, I'm not going to get too sidetracked, as I said, into the results of this pilot study itself, although they were promising in some ways and quite interesting. But what's important to note is that leaving aside the mutated items, which are the distributions in orange and yellow, certain baselines from the literature were met, which gave me more confidence in further analysis that I did after this baseline was met, which is to say that expectations from the literature would be that for a syllable with a voiceless stop onset, you'd expect the p-center to be at its earliest, compared to a sonorant onset, where you'd expect the p-center to be at its latest, and a voiced stop onset, in which you'd expect the p-center to be somewhere in the middle of the two. And this is met if you look going left to right, you have an earlier p-center, a middling p-center somewhere in the middle of the closure, and a later p-center, all with relatively normal distributions across the participants. So armed with this and feeling fairly confident, I designed my second iteration of this task. The frame was kept largely the same. I had a metronome beat at the same speed. I increased the number of trials per item in order to get better bang for my buck out of each item, not least because recruiting participants in the field is sometimes a bit trying. So I was trying to get as much data as I could. The most important thing was that I restricted my stimuli from the wider range and structurally varied set that I had in the pilot study to only the simple possessive phrases, which had turned out to be the most amenable to analysis in the pilot study. To this, I added a set also of simple possessive phrases comprising short vowel syllables, or disyllables rather, which had to be disyllabic for other structural reasons. But other than that, these comprised also nicely paired homophone pairings, which could be varied for presence or absence of mutation. Unfortunately, despite the fact that I had 7 participants who all treated the task confidently and in the way that I intended them to, at least as far as I could tell at the point of recording, things began to look wrong quite early in analysis. The baseline that I was talking about earlier of a progression of p-centers across p-center location from earlier to later across the 3 different consonant onset consonant types rather, was not present, or at least it wasn't apparently present. Metronome beat location, by which we're assuming is meant to be a proxy for the p-center, was apparently invariant across all conditions for all participants, with very few exceptions and with relatively normal distributions around where they tended to cluster. Unlike in the pilot study, metronome beats were not falling in the onset closure for the target lexical items of interest. Instead, they were preceding the beginning of the possessive particle a, just a simple schwa, by about 100 milliseconds in all conditions. This would have been fine if, well, I say it would have been fine. It would have been all right or acceptable if it just didn't meet my hypotheses, but it wasn't just inconvenient. It was a bit of a red flag, because this is supposed to represent the p-centers of these particular items, but this can't be representing the p-centers of these items. It's just metronome beat location being invariant. So what I suspect happened is that there was a conditioning effect in the pilot study, which was inadvertent but very helpful in focusing participants on the items that I was actually interested in getting the p-centers of, or measurements on the p-centers of, in that the smallest possible target with which they were being presented was simple monosyllabic lexical items. They could have these, or they could have more complicated ones, the datives or the possessives. And these were in variation and pseudo-randomized, so they were constantly switching back and forth between different structural conditions in addition to different mutation conditions. When this was scrubbed in the second iteration of the task, structure was also homogenized. And this was intentional, but what its knock-on effect was not intentional, which is that participants stopped focusing on the target lexical items because they were no longer particularly interesting in it from a structural perspective. And instead they began synchronizing metronome beats because they had been directed specifically only to assign one metronome beat per phrase. They began just aligning their metronome beats with the phrase as a whole, which in practice meant that they were aligning the metronome beats with what I suspect very strongly was the p-center of possessive schwa, which meant that I got quite a lot of data on the p-centers of schwa, which isn't that interesting because it was invariant, and no data on the p-centers of these lexical items with or without mutation. So I got quite a lot of data on the wrong thing. My proposed solution is not particularly complicated. It's just to reinstate the conditioning effect, which I believe was present in the pilot study. And I've consulted with a few colleagues in my own university and elsewhere, and I've reason to be confident in this. It's just to precede the stimulus phrases that I had in the second iteration of the task with a dummy item which matches the structure of the lexical items that I'm interested in. So instead of repeating a paw in time with a metronome beat, participants will now repeat in alternation tha, a paw, or taka, a paka. And in this way, I think it's the best possible shot that I've got in this paradigm of focusing them on these without having to go back to having a wider range of stimuli to focus them on the common element across all the different structural variations. And in this way, I hope to be getting data on the things that I'm interested in rather than on just possessive schwa. Now, whether the data that I get aligns with my hypotheses about how the p-center should or should not be moving around is a completely different question. But I'm looking forward to having at least data on the correct thing as opposed to quite a lot of data on the wrong thing. Unfortunately, the plans to deploy the third iteration of this task were very robustly torpedoed by the COVID-19 pandemic, like so many other things. This was supposed to have been rolled out in spring and then in the summer and then in the autumn, which the jury is still out on, but I suspect it might be a little while before I can go back out into the field, which I suspect is not an uncommon story to people listening in the audience. Nevertheless, I'm hopeful that I might be able to trial this either in person or online before the end of the year, and to have some answers as to whether this proposed solution will work in refocusing my participants on the correct items in this task. So to summarize what I've gone through, I had a metronome synchronization task involving multi-word phrases, which was necessary because of the phenomenon that I was looking at, which has to involve multi-word phrases. In a pilot study with a much wider range of stimuli than was eventually rolled out in the main task, there was a common reference point, which was incidentally the lexical items that I was most interested in getting the p-center measurements from. When this was lost, I ended up accidentally unfocusing my participants on the things that I was interested in, and I only got p-center data on possessive a as opposed to on the interesting items of pa, ba, or ma. I think that participants can be refocused using additional material, that is to say dummy material preceding the items that I'm actually interested in getting data on, and the results of this are yet to be seen, but I'm hopeful. We'll see how it works out. I'd be very happy to get any feedback that anyone has on this, either now in the Q&A or personally, and I'm happy to be contacted after the conference. So Guramagi, thank you very much.
Thank you very much for your presentation. Are there any questions?
I have one question, actually.
Sure.
It was a great presentation, first of all. And I was wondering, why did you have to use the metronome instead of looking at these p-centers that you are studying in just natural production? I don't know if that would be possible.
So your question is whether I'd be looking into p-center location in non-rhythmic production outside of the context of a metronome beat.
Yes, my question is if that is something that you can observe in natural production.
Not to my knowledge. I think there are ways of measuring where the p-center falls, from what I understand, has to involve rhythmic productions of some sort, because they're a very shady phenomenon which is largely tied in with the perception of rhythm. So you have to get your participants to be producing something or listening to something or somehow participating in something which is definitely rhythmic and that they accept to be rhythmic in order to get a baseline as to where they think the P center might be intuitively, and then from there you can work on it. So metronome synchronization is one way, but there are other ways which are more geared towards looking at acoustic correlates at the P center. And then in principle, once you had nailed down what the acoustic correlate of the P center might be, you could then try to extract that from normal speech, but you first have to establish what those correlates would be, is my understanding.
Okay.
But I should stress that I'm not an expert on the P center.
I don't, it's the first time I hear about P centers, right? I'm just, coming from a naive point of view. But, I was wondering if there would be like a more natural way to do it. So I don't know if that could be, I don't know if this is the case in Irish, but maybe through using rhythmic, what is it called? Like tongue twisters, trying to use something that is.
Yeah, I think that would be a very interesting way to go about it. And I have also tried using recordings from these metronome tasks as perceptual data to feed in and then get participant feedback on what they do and don't perceive to be rhythmic, or whether they hear homophones as the same or different based on different metronome beat placements. So there's a few different ways to play around with it, but it's certainly an interesting avenue to look into. Thank you.
Very interesting. Thank you.